Please.
Thank you for a very nice introduction and inviting me to speak here. And it's fantastic to see a great lineup of speakers that are forming part now of our age in plant sciences. I've been fascinated by all of the talks. What I want to tell you today is maybe something a little bit different. I want to move towards plant microbe interactions. And what for me is one of the most interesting parts of the genomes, it's part of the genomes that evolve most rapidly in both the pathogens and the hosts, and control the host pathogen interactions. Now, before I begin, I want to highlight that everything I'm presenting today is done by a fantastic group of people that are our lab. And I'm here putting, it's one of the most recent pictures of our retreats and a couple of alumni whose research I'll highlight. I want to also thank our department, Plant and Microbial Biology, as well as Berkeley's Center for Computational Biology, Innovative Genomics Institutes, and all the funders. Now, what my lab does, we aim to understand natural diversity and biodiversity and the rules that plants and their pathogens use to interact, and especially the rules in plant innate immune system, with the goal of leveraging this diversity to move towards more sustainable and rational designs of immune receptors, so we can have a very rapid cycle between a new pathogen coming in and disease resistance varieties that would be available. And we utilize genome editing, we utilize synthetic biology, and so kind of we're moving from what we're most comfortable with and have been doing for a number of years, which is comparative genomics, and you see most of the publications are in that area, and that's what I'm going to also focus on for the most part, towards this now exciting, really new area that's been enabled by rapid, low-cost DNA synthesis and synthetic biology. All right, so the really interesting paradox that has been fascinating me since my PhD is that most organisms do not have antibody-based immunity. Most organisms do have innate immunity, innate immune receptors, which are sufficient for them, for plants, fungi, 95% of animals, bacteria, to withstand diseases. And so my question has been, how do these organisms cope with infections with the same types of rapidly evolving pathogens that infect us? And I've been focusing on two sides of the story. On the disease-causing molecules that are encoded in the pathogens, they're called effectors. And pathogens encode, regardless of which kingdom they come from, they can encode hundreds to thousands of secreted proteins that they inject into the plant to cause disease. Those are rapidly evolving proteins. Those genes, a lot of times, they don't look like anything else in the genome or across genomes. And in plants, we've been particularly focused on the type of immune receptors called NLRs. Those are immune receptors that are conserved across kingdoms. They're present in us, in fungi, even in bacteria. And in plants, they are sufficient on the population level to recognize all of these rapidly evolving effectors. And so the first part of the story that I want to talk to you about is, how do we make sense of these disease-causing molecules, of the pathogens? How do we make sense of what the plants see in their environment when they encounter the pathogens? And one lesson I learned from working in this field, that nothing in evolution makes sense except in the light of structural biology. So very slowly for the past 20 years, we've been, as a community, solving experimentally structures of these effectors. And I'm marking here a publication from my PhD where 2 PhD students, 6 years, we solved the structure of 1 effector, right? I just told you that pathogens encode hundreds and thousands of these effectors. And so it seemed like it was really an impossible task for us to make sense of all of them. Except that a couple of years ago, there's been a major breakthrough. And in computational biology, and especially computational structural biology, and that was enabled by machine learning algorithms that now allow us to predict structures from the genomes. And I want to highlight here that in my lab, this advancement in the field was highlighted to me by a PhD student, Kunyang Seong, who came to me in 2020 before AlphaFold was released. And she's been saying, there's been an assessment, critical assessment on all of the programs, and we now have a breakthrough. These programs are robust enough to take sequences and predict structures. Not all of them will be correct, but we can sort through them, and we can do now computational structural genomics. And that it turned out that Kunyang was absolutely correct. He took, at the time, publicly available software developed by David Baker's lab, as well as others called TI Rosetta. And so before the AlphaFold and other softwares were released, we were in a prime position to test this on the plant pathogen encoded infectors. And so what Kunyang did, she said, okay, well, I looked at the reviews, and there's a review called Top 10 Plant Pathogens. So everyone here in the audience who writes those reviews, please continue writing them. Those are really important. So Kunyang said, well, the top plant pathogen in the review is Magnaporthia rhiza, wheat and rice blast. So let's start with that. And so he took secreted proteins, predicted secreting proteins from this very important and devastating plant pathogen. It's about over 1,000 of these. And he predicted structures of all of them. And indeed, what he was able to show that among sequence unrelated effectors, there have been many structurally related groups. So we could use this computational prediction and structural information to sort through these effectors and group them based on structural similarity. This paper was published in 2021. Kunyang won the Best Graduate Student Paper Award for that one. So I'm really happy he was recognized, his work was recognized for this. But then he moves on and said, like, well, if we can do one, let's do comparative genomics now using this structural information. And then he took a couple of dozens of devastating plant pathogens. He also included non-pathogen for comparison, and he predicted all of their structures to over 24,000 structures, and he did this comparison now across species. And what he was able to show for the first time is that overall, these effectors that do not have sequence similarity often, they're not coming from anywhere. They are a lot of them share similarity to secreted proteins in non-pathogens. And a lot of cases, what we see is a very rapid expansion and divergent evolution. So we start to shed light where those disease-causing molecules effectors are coming from. And to give you a couple of examples, we have a representation of sequence-related clusters, and they're linked with gray lines by structural relatedness. And we have one of these that's conserved across fungi, pathogens, non-pathogens, a few of these actually. And then in a particular devastating pathogens called rust, we have a very rapid expansion of these structural fold, and we have very rapid diversification. So what we think is happening a lot of times in pathogens, there's divergent evolution and then a specific pathogens utilize this fold to enable colonization of plants and manipulation of their hosts. There's another example of classic example of Histulago medicine. It was known that there is a region in the genome here, delineated here, that was critical for virulence. If that region is deleted, the fungus becomes non-virulent, and it was thought that it contained it was just a hotspot for unrelated virulence-causing genes. You can see the sequence similarity groups are here in different colors. What Kun-Yang showed, they're all part of one large structural family. So indeed, it's a cluster, but it's a cluster of a sequence, unrelated but structurally related genes that likely originated through duplication and divergence. There's a lot more analysis in this paper. I'll stop here because I want to tell you more about our plant work. But what it enables us to think is that if non-pathogenic ancestors became pathogens through very large expansion and diversification of specific proteins, and we still see the structural similarity, maybe we can be really rational as well. How do we pick what effectors to go after when we think about plant immune system, and how can we use this information to design a more robust and a better disease-resistant genes? So now I want to switch and talk mostly about this plant immune receptors. And one of the basic principles of innate immunity is that unlike antibodies where the diversity of receptors is generated within a single organism each time, here the diversity is generated and maintained on the population level. So when the pathogen evolves, there is selection under the pathogen pressure for new alleles among this diversity. And because these receptors are really repetitive, they're evolving so fast, for a while it's been really hard to study them. And what we've done, we came up with several new tools for robust analysis that allows us to detect a lot of biological signals in those receptors. And so what we've built phylogenetics combined with structural modeling to examine pan-genomes, diversity in these immune receptors, which are not dependent on any particular reference strain. And we've also applied sequence diversity analysis that haven't been applied before to innate immune receptors, but they're often used for antibody diversity to actually detect and predict rapidly evolving ligand binding sites in the antibodies. So we've applied it now to NLRs. And we've published this work for Arabidopsis, and now applied it and preprinted it for a number of crop species as well. What this analysis showed us is that in plants, there are two types of the receptors. One is they're generally conserved, or they include very few alleles in the population that can be maintained in the population. We call them non-highly variable receptors, or NLRs. And so here's this entropy or sequence diversity signal for one of these classic genes. And there's another subset of the receptors that exhibit extreme levels of polymorphisms in the population. Moreover, they exhibit this really high entropy in their part of their loose-enriched repeats that are known for effector binding. Now, previously, it's mostly thought that this is the only result of the selection. And I completely agree. There is a very strong diversifying selection acting on this. But what we also started to wonder is, do these two types of receptors have genomic features that are differentiating them? How this diversity versus no diversity can be maintained or generated in the populations? And Chandler, who's a grad student in the lab, she preprinted a really fantastic analysis of these two groups, and she showed that indeed there are several genomic features, including transcription level and epigenomic features, as well as distance proximity to transposable elements that statistically significantly distinguish these two groups. So we now start to think that there might be mechanisms to maintain or generate diversity on a population level to enable rapid pathogen recognition, right? The other bit that we've learned from these types of analysis is that we can use sequence information to predict these ligand binding sites in this highly variable immune receptor. So population level diversity is predictive of the binding sites. And we were able to validate this using a structure, experimentally solved structure that became recently available, several of them actually. And we can show that using computational methods without any knowledge about the real structure and using only predicted structures, we can recover over 80% of amino acids that are actually involved in the ligand binding site. And that's about as good of a precision and recall that are currently done in antibody and are used in antibody engineering. So that was fantastic because it led us to think that we can start using this information to change the specificity of immune receptors and engineer them. And why this is critical right now, it's critical for a couple of reasons. First is by domestication and modern methods of agriculture that we use, we really narrowed the pool of genetic diversity in our crops. And moreover, we compromised this one of the basic principles of functional innate immune system that needs the diversity on a population level, yet we're planting mostly monocultures and genetically identical fields and fields of crops that then succumb to disease. The second reason why I'm really excited to accelerate translation from understanding the basic principles to engineering that was relevant for me personally as a Russian American was the recent war that's been started. And I really want to applaud two of my colleagues that immediately drew attention to the ripple effect and impacts that this will have on the whole world, including our agriculture. So I encourage you to read, especially Alison Bentley's, who's now director of one of the programs at CIMMYT. And what I mean, what can we do? So currently our current status is we're breeding natural diversity, we're taking immune receptors, we're incorporating them in our crops. Typically, that takes 10 to 15 years. With synthetic biology and rational design, I think we can reduce the cycle to maybe 1 or 2 years. And as we've seen with the pandemics, this is critical. This level of expedited immunotherapy for plants, I think, it would be critical for making more sustainable disease resistance. Now, one of the case studies that we've used here is in wheat, and modern wheat that's grown. It utilizes 2 disease-resistant genes that have been introgressed. One of them comes from rye. Another one comes from Aegis lobstauchi, so a wild relative of wheat. And both of these are used in different breeding programs. Now, we've shown that these 2 genes that are really different from each other and belong to different homology groups, they're both those highly variable NLRs. If we look at the sequence diversity, especially across wild species, and we can utilize our computational modeling to predict ligand binding sites of these receptors. And moreover, what a postdoc in Taborsky together with the graduate student Kun Young shown is that they could detect and depict only 12 amino acids that were sufficient to convert one of these NLRs, its recognition specificity for another NLR effector. There's been a lot of more than I'm showing here in the picture that's involved. These immune complexes are maintained in a very highly regulated manner where misregulation results in cell death. But through the structural modeling together with the synthetic biology and building, testing, and refining, they were able to basically make a new designer a receptor. Why I think it's particularly important right now? Well, if we can introduce minimal modifications, and here we've gone down to 12 amino acids, our most recent work with structural modeling, we were able to pinpoint 1 or 2 sites where we can resurrect the defeated NLR genes. We can then move towards editing this variation into the crops. And with the current regulation, I'm hoping that this might not even be transgenic. All right, in the last 1 minute, what I want to talk about as well, that thinking about biodiversity, it's really important to go beyond the street lamps. And this work that we've done on looking at the biodiversity of NLRs was really fundamentally done by one of the first PhD students in the lab, Aaron Bax. And what Aaron asked is, well, we think of NLRs as really the most important genes that can save the plants, but are there any plants that survive without them? And she found that duckweeds, flowering plants that went back to water, they actually lost most of the NLRs. And they're clonally propagating, they're floating in the soup of bacteria, so what do they do? And what Aaron showed that while duckweeds and a few other organisms, they can survive without NLRs, duckweeds in particular, they have an expanded antimicrobial peptide protein families that are evolving very, very rapidly. So we think that in the biodiversity, there might be additional avenues, additional ways that to explore how we can save our crops. So with that, I want to put out a few take-home messages. I showed you that pathogen effector biodiversity reveals shared and expanded structural folds, and I think we can use this information for rational design of immune receptors. In nature, plants rely on multiple mechanisms on how to combat the pathogen, and I think it's really exciting. We're currently just bound by our imagination of which genes we can synthesize to move towards more rational designs. And I want to thank the lab again, especially the work done by Kunyang and Chandler and recent alums, Aaron and Yanina, and collaborators. And I'm happy to take your questions.
Okay, thank you, Dr. Krasilava, for sharing your nice work. Okay, is there any question from the committee members, please?
Yes, I would like to come to this last part. I find that quite fascinating what you told us about duckweed. So there's only one immune receptor, but then there are these antimicrobial peptides. So I just wonder, how specific are these peptides? Is it known how they act? And do you think there is a bit of a limit in transferring things to land plants, to crops? Because, you know, we also hear these talks about the benefits of the microbiome for plants. So maybe they have to balance repelling them, you know, with attracting them. And so how specific are these?
So this is fantastic. This is a fantastic question. I had a version of this talk where I talked mostly about the duckweeds and the 3 papers we published on that. So the answers to your question is, it's super exciting that there's an expansion of this antimicrobial peptides. There's 0 of these genes in Arabidopsis. there's one, and it's been characterized first in macadamia nut, and then in duckweed it's 50 or 60, and they're really rapidly expanding. So they're divergent as rapidly as the effectors I've just told you about, right? So I think there is a really great potential here. And our RNA-seq data, so what Erin done, she established actually a novel pathosystem using duckweed and Pseudomonas pathogens in the lab. She showed that these antimicrobial peptides are upregulated in transcription upon infection. We've synthesized a few of them as well. And so I think there's a really good chance to explore these. To answer your second part of the question, this is not a full story for duckweed. We actually went to the environment, sampled duckweed and the water that it lives in, and we showed that there is a microbiome effect. Specifically, there's Pseudomonas antagonism, and so duckweed's by losing this immune components, we think it gained the ability as well to enable the growth of close relatives of the pathogens that engage in antimicrobial warfare. And so these antimicrobial peptides, while they might be important for direct pathogen control, they might be even more important for selection of the microbiome, of which ones are resistant to them and can grow, right? And which ones are not. So it's very
But how do you know that they're antimicrobial?
They're predicted.
Oh, okay.
There's members of this family that have antimicrobial effect, and that's been shown experimentally.
Okay.
And in duckweed, that protein family is expanded and upregulated upon pathogen infection, bacterial infection.
Thank you.